Well, so I would like to first thank you all for having me here today and for giving me the opportunity to present some of the work that we do in my lab and also in the Genome Editing Center. So today I would like to take a few minutes to actually introduce myself and tell you a little bit more about my education, my training, and perhaps more importantly, my experience with genome editing in engineering. In the second part of my presentation, I will provide you with an overview of the Indiana University Genome Editing Center. I'll describe the various services the core has to offer and the recent technological development that we've been working on as well over the past year or so. Then I will wrap up my presentation by telling you a little bit more about our research program, which deals with rare pediatric neurodegenerative conditions and how we use mouse genetics essentially to address most of our questions. And hopefully that's going to give you some ideas as to how you can use the core and how you can use mouse genetics or genome editing in general for your own research. This research program is also used to develop new technologies that we can later on apply to or offer as a core as a service by our core facility. Just want to mention that my presentation will last about 35 to 40 minutes, and so feel free to interrupt me at any time if you have questions. There's no it's better when there's a conversation than it's just me presenting some data. All right, so before I get started, actually, I'm going to have to move this a little. Before I get started, I would like to thank the people who participated in the work that we'll be presenting today. These include members of the Indiana University Genome Editing Centers, former lab members, in particular, Sébastien Gingras, who's been instrumental in implementing CRISPR technology back when I was at St. Jude. I would like to thank Judy Hallett from the Purdue Transgenic Core facility, who has been very helpful in engineering some of or micro-injecting for us to engineer some mouse models. I would like to thank people from the St. Jude Children's Research Hospital Transgenic Core facility. Yeah, all these people that I've been working with them for years, and they've been instrumental in generating more than 200 mouse models, and you'll see I'll talk about this a little bit in a minute. I would also like to thank our 80 core users from 8 different institutions. Without them, there would be no core, I guess. And finally, I would like to thank the funding agencies, the Indiana University School of Medicine through the SRI, or I can't remember. Oh yeah, the Strategic Research Initiative. I would like to thank the Department of Medical and Molecular Genetics, as well as CTSI for some of the fundings through grant applications by PIs, and also the NIH for some of their support. So for those who don't know me, which I guess there's some of you out there, my name is Stéphane Pelletier. I am originally from Quebec City in the province of Quebec, which is the French-speaking part of Canada. I have a bachelor's degree in biochemistry from the University of Sherbrooke. I also have a master's degree in pharmacology from that same university. I then moved to Montreal, and also in the province of Quebec, where I obtained my a PhD in pharmacology. In 2003, I actually moved to Memphis, Tennessee, to join the lab of James Eiley, who was at the time chair of the Department of Biochemistry at St. Jude and Howard Hughes Medical Institute investigator. And it's in this lab that I learned most of what I know about gene targeting in ES cells and gene knockout and mouse genetics. In 2009, I joined the Department of Immunology, also at St. Jude, to establish the embryonic stem cell and genome editing facility, which was a small genome editing or gene targeting facility that helped investigators with engineering mouse models, especially for the Department of Immunology, but we did, as you will see, help a lot more people in many different departments. So as head of this facility, I designed and oversaw the generation of hundreds of genetically modified mouse models and cell lines. I pioneered the implementation of CRISPR-Cas9 technology for mouse genome engineering at St. Jude. That was back in 2013 when it just came out. And we produced since more than 200 mouse models with this technology. We developed new CRISPR-Cas9 systems for genome editing in cell lines. I established collaborations with more than 30 research groups at St. Jude and across the United States, Canada, and United Kingdom. We helped implement CRISPR technology for mouse genome editing in 4 institutions in the United States and Canada, including the University of Montreal, where I actually graduated from, and the Scripps in California, and I think University of Pittsburgh, and now I'm missing one, I think in Florida somewhere. Anyway, the point is that we've been very busy. We've been helping everybody with developing this technology or implementing the technology. I also supervise the work of more than 30 postdocs, staff scientists, and lab technicians over the last the last 20 years since I moved to Memphis, Tennessee. Here, I just wanted to mention that that if you Google my name, you may not find that we have published these 200 mouse models. Actually, the majority of the mouse models that we've engineered over the past 10, 15 years have not yet been published. Many of them were central to securing funding from the NIH and the Howard Hughes Medical Institute and many other funding organizations. Some of these, though, have been published and have been central to studies that were published in top-tier journals, including Nature Genetics, Cell, and many others. So just to give you an idea of the power sometimes of genetics, this is a good example. All of these papers would not have been published without a genetic model, whether it's a mouse model or a cell line model. As director of this facility, I was also given the opportunity to continue some of the research that I initiated as a postdoc. So I led a small research group that studied rare pediatric neurodegenerative disorders, like I mentioned earlier in the presentation. And also, as mentioned previously, we have used this research to develop and implement many of the genome editing strategies and technologies that we're still using today, and that I brought with me to the core to IU. In July of 2020, I moved to Indianapolis and to establish the Indiana University Genome Editing Center and to continue my own research program on rare pediatric neurological disorders. So the Indiana University Genome Editing Center is a IUSM and CTSI designated core facility whose mission is to provide Indiana University and external investigators access to state-of-the-art genome editing technologies by assisting with the generation of genetically modified organisms in a time and cost-effective manner. The core was established about 1.5 years ago in July 2020, but became really functional by January 2021. We are located on the 2nd floor of the Medical Research and Library Building, which is on Walnut Street in front of the R3, R4 building. And we are in the Department of Medical and Molecular Genetics. The core occupies about 1,700 square foot of lab space and 400 square foot of mouse holding space. And the core is outfitted with all the necessary equipment that is needed to perform molecular biology, cell biology, biochemistry, tissue culture, microinjections, and many others, and many other techniques or approaches. We can also, since maybe 6 months ago, we can also now perform our own sequencing, so that helps with the turnaround time for the generation of different cell line projects and mouse models. So the core currently employs 5 full-time employees: Hanyin Chen, Annelise Cassidy, Disney Thomas, Brendan Daly, and Max Tubin. Anion, Annelies, and Disney are essentially working on the generation of mouse models and traditional transgenic services. And Annelies also works on the generation of cell line models together with Brendan and Max Tubin. So the core offers essentially three categories of services. We offer model generation services. That includes mouse models, cell line models, and we are also working towards maybe engineering some rat models as well. For the cell line models right now, we offer services for transform cell lines, but we are right now working on developing procedures for engineering of human iPS cells and human embryonic stem cells. The core offers traditional transgenic services, which were the ones offered by the previous transgenic core. And finally, we can also work with investigators to develop new technologies. And I'll give you an example of that in a minute. So for the mouse model generation services, these services include everything from the design of the allele to the delivery of genetically vetted animal models to investigators. The price tag associated with these services vary from $7,500 to $30,000, depending on the complexity of the allele that we try to engineer, and as well as the technology that we use to engineer the model. So if we have to go through engineering embryonic stem cells first, this is a little bit more expensive, and that explains the price tag. Our timeline for the generation of these mouse models varies between 9 months and 18 months, again, depending on the complexity of the allele that we try to engineer. So for the cell line model generation services, these include everything from the design of the allele to the delivery of fully vetted clones to investigators. We do not limit the number of clones that we deliver to investigators. We're going to give the investigators all the clones that they would like to have, whether even if the clones are heterozygous or they have bizarre mutations. Usually we screen them very carefully, and we can propose to investigators maybe that there's another clone in the batch of clones that we generated that might be interesting for his research. So we communicate that with investigators. So we do not limit the number of clones that we are engineering for investigators. We also offer a cell line optimization service, which is this is in the case where if the core has never used this cell line before, the service includes testing the clonability of the parental line, the identification of electroporation conditions, and whether the parental line is amenable to homologous recombination, which is essential for the insertion of specific mutations. The price tags associated with these services vary from $2,500 for the cell line optimization to $20,000. Again, that depends on the complexity of the allele that we try to engineer and the success rate of engineering those alleles. Our timeline for the generation of these cell line models is anywhere between 3 to 6 months. We aim for 3, but sometimes it can actually go even. It might take sometimes a little longer than 6 months to engineer these models, and we will keep you updated throughout the process. So virtually any kind of mutation can be engineered by the core. These include insertion of missense or nonsense mutations, which are often used to inactivate genes. Insertion of small or large DNA elements like epitope tags, small digrons, recombinase fluorescent proteins, and so forth, which are usually used as markers or as biological tool. We can also remove large DNA segments. This is usually used to inactivate genes by inserting, if you will, a large deletion. We can also invert genetic material. This is often used to reorganize or rearrange genes so that the genes are in the same orientation in the mouse, for example, as it is in humans, so that we can actually do this inversion and then generate chromosomal translocation, which are often seen in human cancers. So we can mimic this in a mouse, for example. And I do have an example of this at the very end of my presentation. If you're interested, I can show you what that looks like. And finally, like I just mentioned, those chromosomal translocation. This is very interesting. Okay, so the core uses various technologies for the generation of these models. For the mouse models, the core will primarily use CRISPR-Cas9 technology, either in the form of in vitro transcribed RNAs encoding the guide RNA and Cas9 protein, or ribonucleoprotein complexes that includes the sgRNA and the Cas9 protein itself. We can also use transgenesis using conventional mammalian expressing vectors or lentiviral vectors. And we can also use CRISPR-mediated or conventional gene targeting in ES cells if we do not succeed at engineering the mouse model using zygote microinjection of CRISPR reagents. For the cell line models, we also use most of the time CRISPR-Cas9 technology. We can using conventional plasmids, but we're also equipped to use lentiviral vectors if the cell line that you want to engineer is a little bit more complicated to transduce. We can also use transgenesis using conventional or viral-based vectors. So the core, like I mentioned, offers traditional core services. These services include in vitro fertilization, sperm cryopreservation, embryo cryopreservation, pronuclear injection, cryostorage, and all of these services go from $77 a year for the cryo storage to about $6,000 for a round of microinjection. But these are standalone services. These were the services that were offered by the former transgenic core. We kept those because some investigators want to prepare their own targeting constructs and everything. So we still offer microinjection as a service. Yeah. So since its inception, the core has completed more than 50 model generation services. These include cell line models, mouse models, and some of the cell line optimization services that we offer. The core also performed more than 98, this number is probably 120 right now, but more than 100 traditional transgenic services. These include, like I mentioned, microinjections or sperm cryopreservation and embryo line reconstitution by in vitro fertilization and so forth. The core also maintains more than 400 cryopreserved mouse models and cell line clones. All right, so in addition to these conventional services, if you will, the core, our facility essentially can assist with the development and implementation of new technologies. For example, we can assist with genome-wide CRISPR screen if you are interested in that kind of technology or that assay. We can help with the implementation of CRISPR applications beyond genome editing, whether it's to label some genomic regions and so forth and so on. So we can help you with these technologies and understand how we can make this work with you. So one example of the technology development that we currently are being investigating is the use of short artificial intron for the generation of conditional alleles in mice. So this is a project that's being led by Annelise Cassidy together with Leslie Thomas in the lab, and Hanyan Chen as well is involved in this project. So the approach consists of inserting a short 201 nucleotide long DNA cassette encoding a small artificial intron flanked by 2 LOXP sites, which are shown here within the exon of a gene. All right. So this cassette, also known as the AIV4, we use this notation all the time, contains sequences encoding a splice donor, a series of stop codons in all 3 frames, 2 log speed sites that are flanking a branch point, and a polypyrimidine track. And then this is all followed by a splice acceptor at the end. So this little cassette is inserted in the exon of the gene, such that in the absence of the recombinase, the small cassette is functional and is actually functioned as an intron and is removed by the splicing machinery, allowing for the normal expression of the protein. In the presence of the recombinase, the recombination between the 2 log speed sites occurs, and AIV4, the small intron, essentially is crippled. And that results in the inability of the splicing machinery to remove the small intron, leading to premature termination of translation by one of those 3 stop codons here. So the advantages of using these technologies are manifold. The engineering process involves a single insertion event, as opposed to 2 insertion events that must occur in cis, which is normally used by the current strategy. So when we try to engineer conditional alleles right now, we try to insert a log speed site in front of the critical exon, and then the second log speed site downstream of that critical exon. In this case, we use 2 guide RNAs to introduce these 2 log speed sites, and either 2 homology-directed repair templates or a large single-stranded DNA molecule. In the case of the artificial intron, this would involve a single insertion event, and this might reduce or increase the chance of engineering the allele by a factor of 4. Essentially, that's the take-home message here. The technology can be used to inactivate single exon genes as well, whereas we could not do this with the current approaches. The short artificial intron is relatively easy and cheap to produce compared to large single-stranded DNA molecules. And a single round of microinjection, as opposed to 4, may be sufficient to produce the allele. And as you will see in a couple of slides, it is the case. So this strategy was developed by Horizon Genomics back in 2017. It was validated in cell lines, but it's never been tested in mice. And so we wanted to see whether we could use this strategy to engineer more efficiently conditional alleles in mice. So to test this, we use our favorite gene, which is SCYL1, and you'll see I'm going to talk about this a little bit more later. We inserted the AIV4 cassette within exon 3 of SCYL1. The insertion of the cassette was confirmed by PCR genotyping and Sanger sequencing, and mice bearing the insertion were then bred to homozygosity, and the functionality of the allele was assessed by looking at SCY1 expression in the brain of these animals. As you can see here, in these 3 lanes here, SCY1 is expressed to levels that are comparable to the wild type SCY1 mice, suggesting that indeed the small artificial intron is recognized as an intron and spliced out, allowing for the proper expression of the protein. So to test whether inactivation or recombination between the 2 log speed sites would actually cripple the intron and result in the inactivation of the allele, we bred mice with a pre-deleter mouse, as shown here on the left. I don't want to give you the details. The important part is that we were able to engineer mice, as you can see here on the genotyping panel, mice that were homozygous for the deleted intron or the inactivated intron. And importantly, these mice did not express SCYL1. And actually, when we looked at the phenotype of these mice, we also looked at the phenotype of these mice, and they actually developed loss of function phenotypes that are very similar or identical to loss of function phenotypes caused by the conventional deletion or knockout of SCY1. So from these results, we concluded that the short artificial introns can be used to engineer conditional alleles in mice. Insertion occurs in about 15% of edited mice obtained from zygote injections, which suggests that a single round of microinjection may be sufficient to produce the mouse models with conditional alleles, whereas normally it would take anywhere between 2 to sometimes 6 rounds of injections just to obtain LOXP sites alleles with 2 LOXP sites on the same allele. So this may significantly reduce the cost of making these mouse models, and this is what we're hoping right now. So right now we're also testing the universality of the approach. So we have designed 4 or 5 more mouse models using this approach, and we are, as we speak, engineering those, producing those. We would like also to test whether other recombination or recombinase-based system could be used to engineer conditional alleles. We're also trying to find ways to engineer conditional point mutation using a similar technology. And we think that, and we hope this is going to work out really well. We also hope to implement this technology, at least the artificial intron for the generation of conditional alleles in mice. We would like to offer this approach as a standard approach for the generation of conditional alleles in mice. So this is what the core has been working on as well. So at this point, I think that if anybody has questions about the core and services we have to offer or anything else, that might be a good time, because at this point I'd like to shift gears a little bit and would like to tell you a little bit more about our research program and how we use mouse genetics to gain insights into disease mechanisms. So any questions?
So we had one question in the chat.
Okay.
About the use of the short artificial intron. So is this available currently? And if so, do you have a cost for, the question is, one-step conditional allele generation? Yeah,
so we are not offering this right now as a service because we want to make sure that this is going to be universally applied and that we can really use this technology and we want to make sure that when we're going to engineer models with this technology, that we will be able to, that the allele will actually behave as intended. So we are trying to engineer 4 or 5 more mouse models to confirm this. And as soon as we get data from these models, then we're going to start offering the service. This will reduce the cost of making the mouse models using with conditional alleles. We do not have a price tag as right now because we don't have enough data about the success rate of engineering those alleles, but it looks very promising. So it's probably going to reduce the cost of making these models. Right now we charge $24,000 to engineer a mouse model with a conditional allele, and I'm expecting to reduce the cost of making those to maybe 16 or something like that. So, but can't promise you anything. I have to run this by my financial officer. But anyway, yeah. So we hope to be able to offer the service within a year.
Great. Does anyone else have questions? If you want to raise your hand, or you can just unmute yourself and ask, that'd be fine.
That's good.
Yeah, I don't see any more in the chat. Okay. Maybe people can ask again at the end.
Yes, absolutely. Yeah. Okay. So as I told you at the beginning of the talk, my lab is also interested in understanding the mechanism underlying SCY1 deficiency syndrome, which is a rare pediatric neurodegenerative condition. So SCY1 deficiency syndrome is characterized by recurrent episodes of liver failure, which in some cases require liver transplant. Global postnatal developmental delay, spine and chest abnormalities, including kyphosis, lordosis, scoliosis, uneven shoulders, and bell-shaped chest and abdomen. Joint laxity, respiratory phenotypes, including bronchiolitis, pneumonia, and upper respiratory tract infection, which in some cases required hospitalization. Most importantly, perhaps, is the neurological phenotypes that we see in those patients, which include cognitive defects, ataxia, tremor, stutter, muscle weakness and atrophy, and motor dysfunction, leading to partial paralysis of their legs and arms. The syndrome affects people from any or various ethnicities. We I found people with SCY1 deficiency syndrome, German population, the British population, Cuban, Pakistani, Turkish, Italian, Chinese, and as well as two kids in Canada. And I've been involved in the characterization of their genetic mutation. The life expectancy of these individuals is unknown, but based on some of the mouse work that we have done, we suspect that they might die prematurely, probably in their mid-30s. So the syndrome is caused by homozygous or compound heterozygous mutations, most of which are frame shift mutations, leading to the inactivation of the allele. Other mutations include the missense mutations and microdeletion, which affects the stability of the protein. So what is SCY1? Essentially, it's a evolutionarily conserved protein with orthologs from yeast to human. The protein is ubiquitously expressed in mouse tissues and as well as human tissues, and localizes to the ER Golgi intermediate compartment, as you can see here in this punctated pattern of distribution, and which is actually a site of concentration, folding, and quality control of newly synthesized proteins. So the protein is characterized by a N-terminal protein kinase domain. Centrally localized heat repeats, as you can see here in red, a proline-rich region, and a colloidal domain located at almost the very C-terminus of the protein. The C-terminal segment here is overall pretty disorganized, as you can see here in the three-dimensional structure of SCY1. The protein kinase domain is devoid of catalytic activity due to mutations in the amino acids that are have been shown to be essential in other kinases to be active. So the protein is considered to be a pseudoenzyme or pseudokinase and may function as a scaffold linking signaling molecules together, or as an allosteric regulator of other kinases. Several essential functions have been proposed for SCY1. These include the facilitation of nucleocytoplasmic shuttling of tRNAs via interactions with components of the nuclear pore complex exporting T and tRNAs, the regulation of protein turnover via the formation of molecular complex, a molecular complex containing Hex14 and pol-like kinases, the transcriptional activation of pol-B and TERT expression via the direct binding of SCY1 to these to their promoters, or the regulation of CUP1 mediated retrograde transport of ER resident proteins via the interaction of SCY1 with components of the CUP1 complex and R4 and GORAD, which is a small Golgi. However, whether these functions are really regulated by SCY1 is currently debated, in particular by myself. And more importantly, at least for me, is whether deregulation of these pathways or these functions actually contribute to disease pathogenesis is unknown. As of right now, the consensus is that SCY1, or in the absence of SCY1, CUP1 mediated retrograde transport is affected in the patients, and that's why it's causing the disease. So to actually better understand the function of SCY1 and identify molecular mechanisms underlying SCY1 deficiency syndrome, we decided to identify molecular interactions that are essential for function. And to do this, we actually engineered an allelic series in mice by modifying the endogenous SCY1 locus using CRISPR-Cas9 technology, and assess whether the mice would develop loss of function phenotype, which include, as you will see in a minute, growth retardation, muscle weakness, and motor dysfunction. So we use the mouse because the SCY1 knockout mouse actually developed most of the phenotypic changes that we see in patients with SCY1 deficiency syndrome. These include postnatal development delay, which are clearly visible by 4 to 8 weeks of age, chest and spine abnormalities, including scoliosis, lordosis, and kyphosis, and motor dysfunction, as you can see here in the video on the right, which results in a complete paralysis of the hind limbs and weakness of the forelimbs by 10 to 12 weeks of age. And these mice die usually around 8 months of age. So there's a premature, they die early. So today we engineered 12 mouse models using CRISPR-Cas9 technologies. These mouse models encode various mutant forms of SCY1, and we did this by modifying the endogenous SCY1 locus. This is just to see, I think that's it. Yeah, it's 12. So we engineered those 12 mouse models, and we engineered those mutations based on previous studies in our lab and others that showed that deletion, for example, of the last 10 amino acid of SCY1 was sufficient to disrupt or prevent interaction between SCY1 and components of the COP1 complex. The deletion of the coil domain was sufficient to prevent interaction between SCY1 and DDX1 and FAN98A, which are RNA binding proteins found in cytosolic stress granules, which are also implicated in various aspects of RNA metabolism. The deletion of amino acids 704 to 743 prevented the interaction between SCY1 and components of the outer shell of the COP2 complex, which is involved in ER-Golgi physical transport. We also removed the protein-rich region of SCY1 to see whether there would be an effect on the function of the protein. We also engineer mice with a N-terminal flag tag. And although we engineer that mouse for other reasons, we realized quickly that actually introducing an internal flag tag to SCY1 led to the mislocalization of the protein and prevented membrane lipid binding as well. So you'll see the impact of this in a minute. We also engineer many different mouse models with missense mutation that prevented the interaction between SCY1 and GORAB. GORAB is a Golgi localized coil protein that may function like a Golgi by tethering cytoskeletal or membrane elements to the Golgi apparatus. We also engineer mice lacking the 5 of the 6 heat repeats in the middle of SCY1. And this we know can prevent SCY1 homooligomerization and or tRNA binding. And lastly, we also engineer mice lacking the entire C-terminal segment of SCY1 by removing its entire C-terminal domain, which prevented interaction with the vast majority of the proteins that I just described, and also 2 other proteins, one other protein, SMD1, which is part of the RISC complex. And this C-terminal segment of SCY1 is also the one that was shown to bind directly to the promoter of Olby and TERT. So we engineered these mice, we bred them to homozygosity, and what we found was a little bit surprising and unexpected. Unlike SCY1 knockout mice, which showed growth retardation, motor dysfunction, and muscle weakness, as you can see on these 3 graphs here. SCY1 knockout. Mice expressing mutant forms of SCY1 that actually prevented interactions with many of the protein complexes involved in protein trafficking, RNA metabolism, and transcription showed absolutely no overt abnormalities. Only mice lacking the heat repeats or the mice with the internal flag tag actually showed a loss of function phenotype. For us, that was absolutely spectacular. I really thought that the regulation of COP1 and so on and so on was key to disease pathogenesis, or the deregulation of COP1 function was key to pathogenesis in this case, and it is not the case. So what we found is that, what we concluded from this is that although SCY1 can interact with several protein complexes known to regulate housekeeping functions, such as membrane protein trafficking, RNA metabolism, transcription, none of these interactions are actually required or essential for SCY1 function in vivo. We found that localization of SCY1 to membrane structures along the secretory pathway is critical for function. We also found that oligomerization of SCY1 may play an important role for the function of SCY1 in vivo and supports the view, and our data supports the view that defective lipid and or tRNA trafficking or metabolism may be a major disease mechanism in SCY1 deficiency syndrome, and not the regulation of protein trafficking via interaction with couple complex and other protein complexes. So I think that these results illustrate the power of allelic series in mice to identify key molecular interactions that are essential for function. And we are currently investigating the role of SCY1 in regulating tRNA metabolism and trafficking, as well as lipid trafficking and metabolism. We are also exploring right now the therapeutic potential of gene replacement for SCY1 deficiency syndrome, and we'll do this by engineering, of course, a few more mouse models. All right, so with this, I just wanted to conclude my presentation with this. I just wanted to give you a little bit of an idea of how we can use mouse genetics and to gain insights into disease mechanisms. Clearly, these studies are not, we're not done with them, but I just wanted to give you an idea of what the core can do and what we can do for you, and how you could incorporate this kind of studies in your research. I would like to thank you all. Thank you all for your attention. I just want to tell you that if you would like to have more information about the core, you can visit our website, which is on the IUSM website. Our webpage on the IUSM website. You can also get in touch directly with me, Alex Cassidy or Henry Chen, and probably anybody from the core. So with this, I'm gonna stop here, and I would be happy to take any questions from you. Thank you.
Great, thank you, Stefan. There's a question in the chat. One question, if you compared inserting an artificial intron or editing for a single point mutation, which would have higher editing efficacy and fewer side effects in vivo or in vitro?
What do you mean by side effect? I'm not,
maybe off target. See if you want to unmute yourself and clarify your question. You're welcome to do that.
Okay, I'm not quite sure, but like side effect, I mean, any immune response, like to the human, like the RNA vaccine, they might have some side effect to the human. Some immune response like that way to if in vivo.
Oh, so if these would trigger an immune response, is that what you mean?
Yeah.
Okay,
might be like
so in mice, obviously, when we engineer those mutations, they're going to be germline mutations. So the immune system of the mouse won't be recognizing this as a mutation. If you were to engineer, let's say T cells with these mutations, or any other cell, and you would put that in the mouse, this would be recognized by the mouse as non-self, and you would probably trigger an immune response. When we engineer mouse models with Cas9, for example, Cas9 could trigger an immune response, but we do this at the zygote, at the pronuclear stage zygote. So we microinject those zygotes, and Cas9 is going to be present in these zygotes for maybe a few hours. So it will be expressed and will be degraded. It will do its job, will edit the site, and then will be degraded. So there won't be any major immune response triggered by the administration of Cas9, for example, in zygotes. Does that answer your question?
Yeah, how about the editing efficacy?
Okay, so engineering a non or a missense mutation in mice is more efficient than engineering a conditional allele with an artificial intron. The artificial intron is 201 nucleotide long, and so this reduces, of course, the chances of having this mutation inserted at the precise location. So the missense mutation, we just change a few amino acids usually, and that's it. So the more complicated, the bigger the less chances that you will have to obtain the mouse model essentially. The success rate depends on the complexity of the edit that you try to engineer. Okay, I see the other questions. Should I just read them? Yeah.
Yeah, go ahead.
Okay, so how much does it cost for one step condition? Okay, so we answered that.
Yeah.
The phenotype suggests loss of ciliary function, scoliosis could be one element. Could we send cilia from your affected mouse for EM? Or has it been done? It could be one membrane from. Well, so no, we have not done this. I am not opposed to do it. When we characterized the mice, we really went for the most obvious phenotype, especially when we did this allelic series. We looked for these for growth retardation and motor dysfunction as a readout for the function of the protein. But again, that might be very interesting. This is something that we could look into. To look at the cilia. Yeah. There's another one. Okay, comparing between inserting artificial intron and editing. Okay, so that I just answered that question. The next question is regarding SCY1 project. Have you looked at lower motor neuron degeneration? Do neuromuscular junction degenerate first, for example? Okay, so we think it's a die-back mechanism that's occurring in these mice. So actually, when we looked at the neuromuscular junctions, we looked at 8 weeks of age and they were already gone. So maybe I should have shown some data on this. I didn't want to spend too much time on this. I just wanted to focus on the engineering aspect or the strategies that we're using, but we cannot see neuromuscular junctions on the quarter-strip femoris. So essentially that big muscle on in the leg of the mouse. There's no neuromuscular junctions anymore. The muscles are completely atrophied. There is, so it's completely gone. And we know that by 8 weeks of age, we still have motor neurons in the spinal ventral horn. And these motor neurons, some of them actually show TDP-43 pathology. If you are familiar with ALS, this is one of the major characteristic of ALS motor neurons in ALS patients, so that they have this TDP-43 pathology or relocalization of TDP-43 to cytosolic stress granules, if you want. So we have the exact same phenotype in the SCY1 mice. So we think it's a die-back mechanism that's occurring in these mice, but we don't know. We know very little because we know very little about the function of the protein, which is why I started using this strategy. So that the strategy of engineering analytic series in mice, this is why we decided to do this, because we could not find a function for SCY1 regulating any of the proposed mechanisms or pathways. Okay, next question. Does that answer your question? I hope I did. Maybe. All right. So do you? Yes, thanks. Okay. Okay. Do you engineer producer cell lines? What? I'm not sure what this is. Conrad, can you be a little bit more specific or help me out here? I don't know what the producer cell line is.
Conrad, if you want to unmute yourself, that might be helpful.
Oh, gene therapy applications. Okay. Yes, we can help with this. Right now, we do not offer this as a service, but we can help with this, and we can help. We can also talk to the vector core. I guess there's a vector core that produces viral particles, but I'm not sure exactly what they're doing. They do. I think that Emily Hopewell is working in this lab, but I'm not sure. So Ben, yes. Well, Ben, we can get in touch if this is something you think that might be very interesting. I mean, I'm happy to work with you on the EM stuff. So yeah, I'd be happy to chat with you about that. And okay, I think this is all of the questions for now.
So I can just ask a few more questions if people
Yes.
are generating their own. So if someone wants to do, let's say, a cell line project, what do you recommend as a starting point? Do they need to characterize their own cell line? Do you have some that you already know work and a list that you know don't work, or how should they go about getting started?
Yeah. So we started. When we started the core, I was trying to get as many projects as I could so we can have a feel of what cell line works well with which one don't. So essentially, we also developed a cell line optimization service just to make sure that we will be able to engineer the cell line. So right now, we can engineer hector-93 T cells, mouse embryonic fibroblasts, human fibroblasts. We can engineer we've been able to engineer some MC38 cells that are used sometimes to study tumor formation in mice. We have been able to engineer 143B cells, which are some cancer cell line. We are working on many other ones as well. The starting point would be to get in touch with us, describe get in touch with us, and then we can have a conversation as to what your biological question is so that we can actually engineer the model that will answer or address the question that you have. But yeah, does that answer your question?
Yeah, so I was curious if some cancer cell lines are, have a lot of cytogenetic abnormalities, they have duplications, and does that cause a problem for getting your allele modified if they have multiple copies of a gene?
Yes, it does. It does. So if we are inactivating genes, usually our approaches are efficient enough to inactivate those genes, and the strategies that we're using, we usually can do it even if you have multiple copies of the chromosome or the gene, for example. When we try to engineer homozygous missense mutation, though, it becomes very challenging because we never know how many targets we have. So then it adds to the complexity of the engineering process. It doesn't mean that we cannot. It just means that we might have to screen more clones if it's the case, just to yeah.
Great,
yeah.
Thank you.
But the 288 cell is important. Yeah.
Yeah. And so as people wanting to use to make a transgenic mouse, should they have their own vector, or will you also help in designing the construct?
Right. So for everything that we do in terms of engineering mouse models, we offer services that go from the design of the allele to literally the delivery of the mouse model, fully embedded mouse model. So for transgenic mice, we can do the same thing. We can design, and actually we're working on this. Sometimes instead of using transgenesis, we're going to knock in transgenes at the rosa locus, for example, and this becomes like a large knock-in, if you will. It goes within the category of large knock-in alleles, if you will. But we can help with engineering transgenes using conventional plasmids as well. Yes, we can do this. We don't have a pricing structure for it, and I think that we can do it at cost or something like that.
Okay, are there any other questions? Feel free to unmute yourself and ask if you'd like to. I don't see any more. If there's anything else you'd like to add, Stefan?
No, simply that thanks for being here today, and I hope I gave you an idea of what the core can do and the kind of genetic alterations or mutations we can engineer either in mice or cell line. yeah, just reach out to us if you have any ideas, any projects you would like us to help you with.
Great. Well, thank you so much. Thank you for everyone for coming and for Stefan for a really great seminar. Thank you.
All right, thank you.